So with glycolysis, technically it's a fermentation because there's no oxygen involved. And so we left off thinking about substrate level phosphorylation, which just means it's during the process of a physiological pathway where you have substrates, that have some phosphate that they don't stop substrate level phosphorylation. So you get the glycosylglycerate donating a phosphate off to ADP, and that will form ATP. So it's at the level of the substrate. Well, for glycolysis, so this phosphorylate and phosphorylate. But it depends on the pathway. Like you'll see as we work through the metabolic pathways that there's some options available to microbes on how they phosphorylate ADP, like what substrates are involved. And so with, they had it color coded here where stage 1 is the gold up top, where they actually have to expend ATP. They often say it's like priming the pump. You got to expend a little to get a little. And so by the time that's done, you end up with glyceraldehyde 3-phosphate in stage 2. And that's where you start to see some ATP production. And remember, there's 2 of those G3P molecules, so you get 4 ATPs. And then you have to subtract the 2 that got expended. So you end up with 2 ATP, which isn't a lot, but at least you stay alive. So that's okay. All right, so I think we left off there. And then with stage 3, we're getting into that last bit on the image where they start making fermentation products. And I'll get a little bit more into fermentation in more detail later. But for right now, let's just say, yep, that's the stage where they make fermentation. And it's usually they start with pyruvate. If you look at the normal glycolytic pathway, you go to pyruvate, and then they push different reactions from pyruvate. And if you look at them, and I'll teach you how, yep, those are fermentation reactions that we see going on there as well, just like glycolysis is a fermentation. And so one of the upsides of stage 3, and I'll go back to showing you the figure, is that NAD plus gets generated. And that's good. And so NAD plus links back to stage 2 of glycolysis. And you have to realize that microbes, as well as far as I know, any living organism doesn't really have a ton of NADH to play with. And so they keep kind of recycling, like they'll use NADH and then it goes to NAD plus. Then NAD plus can go and get converted back to NADH. And so I'll show you what I mean here when we look at the diagram. But having that NAD plus generated in stage 3 is useful because it can keep pushing glycolysis going forward and keep going and keep going. With fermentation, we're going to find, and I'll get into this again in more detail later, but we're going to find that the cells, the bacteria, when they make their final products, they don't really care about those. They just excrete them out. That's not their goal. And so to them, it's a waste product. Now, there's a $1 billion industry out there on the waste product of yeast when you think about alcohol production. To them, it's a waste product and it's actually a toxin. Of course, for us, it's a $1 billion industry. So that whole idea of them excreting out these fermentation products, we've certainly capitalized. It's not just alcohol production that we get useful fermentation products in with all the human uses that we have. And I'll get more into that, but that's the one most of us are probably, immediately think about. So let's go back to the diagram. So here you're seeing the idea of stage 3 in green down low, and the idea that you start with the pyruvate, and then you can see that they've shown 3 examples of what could happen to pyruvate during stage 3. So this is just 3 examples. Microbes can do so much more than this. All right, so these are just some examples. And one we're probably most familiar with is the one that goes straight down to ethanol production. But you see, with ethanol production, NAD plus does get made, and it is useful. So if you look at that more closely, NAD plus is getting made, and then it says that goes to stage 2. So if you go back to stage 2 and look a little bit more carefully, you can see NAD plus there. You have this aldehyde phosphate. The electrons that are going to be given to NAD plus, and then that creates, and then that feeds back down to stage 3 to keep stage 3 going. And so you have this kind of recycling going on with the NAD plus to NADH. So once NADH is made during stage 2, it comes down to stage 3, and you can see it's getting used to push those reactions. So I'm going to update these slides. I've added an additional slide that I wanted to kind of spell out what's going on here. Where'd it go? So I will update the slides later this afternoon to include this one. But so here's some jargon for you that all means the same thing. And it depends what class you're in, what textbook you're reading, who is talking to you, what they call it. Energy carriers, electron carriers, reducing agents, reducing molecules, electron donors. That all means the same thing. So what we're talking about are molecules like NADH, NADPH, FADH2. So they're, in the form I've written them, they're fully reduced, which means they've accepted some electrons. And now they can go participate in some reaction that needs electrons. All right, and so their job is to go and donate electrons to some electron acceptor that needs it in order to push a reaction. And so these are really good molecules to form. This is a good goal for a microbe as well as for us. If we can make these, they're useful. They're good amount of energy in the electrons they carry. They help push reactions that we want to go, make them go. And so keep that in mind as you look at these physiological pathways that we start looking at through the next several lectures. If you can make these, if a microbe can make these, that's a good strategy to staying alive, getting your physiological needs met. Okay? So back in glycolysis, we see that, yeah, it does. It makes the, NADH up top in stage 2, and then that can diffuse down to where pyruvate's doing its thing and it can help push those reactions. And then the NAD plus can go back. All right. And so it's a nice little game that they've they're playing in order to keep glycolysis moving forward and keep it going. And of course, the big payoff there is ATP. They're getting their ATP as they do this. But those, I tend to say reducing agents when I talk about NADH, similar molecules, but all those that I listed out on that slide mean the same thing. Okay, so we've gone through stage 3. So let's think about, oh, by the way, there I put in a couple of extra slides. So this is glycolysis as well. This is glycolysis as well. So whatever your brain prefers to look at, that's fine. It's fine. I just tend to teach off this one. I like how it's color coded and I'm just used to it. Okay, one of the things I want you to think about is the pyruvate. So once the microbe has done stage 1 and stage 2, and pyruvate gets produced, that's kind of a decision point by the microbe. So I like to think of a microbe hanging out doing this, and it gets to pyruvate and it puts the brakes on. It sits there and goes, all right, what should I do with this pyruvate? I have some options available depending on the microbe. And depending on where I put pyruvate next, I could get a really good payoff or some level of payoff that'll keep me alive. The some level of payoff would be to keep it going stage 3, because you can get some good molecules from that. And depending on the fermentation pathway, you might actually get a little bit more ATP. So that's not bad, but it's not great. There are better ways to deal with pyruvate if you're the type of microbe that's evolved to do some different physiological reactions that can really make pyruvate shine in a way. All right, so think of pyruvate as a decision point. Imagine the microbe just sitting there going, okay, based on what I have around me and what's going on in me, what can I do with that pyruvate to maximize my payoff? And the payoff is how much I want to get a lot of ATP. That's my payoff. So how do I get as much ATP out of this pyruvate? And if you're a microbe that is floating around, maybe in a river or something, and you're like, oh, I have some oxygen around me, and I use oxygen. That's something I use. So I'm not going to put pyruvate into stage 3 because I have oxygen around me and in me. So instead, I'm going to push pyruvate into the TCA cycle, which is the next one I wanted to talk about, and something that your body does as well. So hopefully you have some vague training on that at some point. So it's also called the citric acid cycle, which I tend to say, that's what I learned when I was first learning this. So if you hear me slip and say that, it's the same thing as the TCA cycle, which your book uses that terminology. So what happens is pyruvate will get shunted to the TCA cycle. And if you look at what's happening to pyruvate, it gets broken all the way down to CO2, oxidized to CO2, which means electrons are taken off of it. If you're oxidizing something, you're stripping electrons off of it. The fact that you can get it all the way to CO2, that's good. That's really good. You maximize your ability to capture as much energy. If you can break a carbon molecule down all the way to CO2, that's awesome. And so that's about as good as it gets. And so that's a good payoff. Then we also see in the TCA cycle that these energy carriers or reducing agents get made, which is good. This is a good thing for the microbe. NADH, FADH2 get made or synthesized. And then during the TCA cycle, we see that ATP, or depending on the microbe, they might make GTP, guanosine triphosphate, which can get converted to ATP really easily. So we pretty much just say ATP gets made during the TCA cycle, and we're good with that. Okay, so the microbe is in an environment with oxygen, and it does glycolysis to pyruvate. It says, I have oxygen. I'm going to send pyruvate to the TCA cycle. So it enters in, TCA, see, I put TCAC for citric acid. It enters into the TCA cycle from glycolysis. And the first thing we see is that you get pyruvate to acetyl-CoA. And then acetyl-CoA will react with oxaloacetate, OAA, and you get citrate. So that's why they call it the citric acid cycle. Citrate's the first stable product. That's often how they name a pathway. They look at the first stable product. So then after that, once citrate gets made, there's a bunch of reactions that follow that. So I just put arrow, arrow, arrow. Yeah, there's a lot of reactions that go from that way. And it's a cycle, so it's going to be a circular pathway. And you'll end up back with OA. I'll show you an image of this in a second. So during this whole thing, what's going on is pyruvate comes over from glycolysis, enters into the TCA cycle, and it gets broken down to CO2. That's a huge thing. So check that box. That's awesome that you can fully break it down to CO2. You've made some energy molecules or reducing agents. You've got some ATP. This, all in all, this is a really nice thing. Now, nowhere have I said anything about oxygen. So you have to hold that thought. It's not quite here that we are thinking about the microbe knows it has access to oxygen. So this is kind of like the intermediate step to the big payoff that comes in. But there's definitely some good payoffs here. And so this is the citric acid cycle. And you're seeing pyruvate at the very top coming in and reacting. First, you're getting kicked off of it, and then you end up reacting with COA, coenzyme A. And then you see the products of CO2, CO2. And then if you just look through that, you can see that CO2 in blue is coming off at a couple different points of the cycle. That's coming from pyruvate. That's a really good thing. Anytime you break it down, any carbon molecule fully to CO2, you maximize your payoff. And then you see citrate being formed. That's the first stable product here, where you have acetyl acetate and acetyl-CoA naphthalene, and you end up with. And then if you follow that through, you can start examining it based on what I just told you about those reducing molecules. You go, oh yeah, there's NADPH. There's SNDH2. Oh yeah, and there's GTP or ATP. So if you start to understand like the basic rules of the game, that microbes want reducing molecules, they want ATP, and if they can break something down all the way to CO2, really good situation. So the TCA cycle is a definite payoff for the microbe. Now, like I said, there's nothing in here so far where you see oxygen, but it's coming. Questions on the TCA cycle? Some of you had this in another class. I know if you're chemistry majors, biochem. But I'm not going to ask you step by step. Like you don't have to memorize the whole thing because we're not in biochemistry. But knowing how it kicks off, pyruvate in particular, I want you to think about other molecules, by the way, can come in to the TCA cycle, not just pyruvate. But for this, what I'm trying to get across now, let's just focus on pyruvate. And then there's a few key things you need to know about the TCA cycle, which we've covered. But you don't have to know all the enzymes and all the intermediates, okay? This is another version of it that I put in case you want to look at it this way. Here's another one with more words on it for studying. And again, you can see they're trying to emphasize, yeah, it's not just pyruvate that can enter into the TCA cycle. But let's just focus on pyruvate for now. So when you look at the TCA cycle, you get CO2 being produced. That's all coming from pyruvate. We say that pyruvate is decarboxylated, means you're cleaving CO2 off of it. And then you have those lovely reducing molecules, NADH, FADH2. You get ATP. I'll talk more about redox reactions, by the way, so we'll get into that. Depending on the microbe, ATP or GTP, but I'm fine if you just say ATP gets made. You don't really need to worry about the GTP. Okay, so all of this is a good payoff. Now, so the microbe has taken pyruvate, put it into the TCA cycle, ran the TCA cycle, and now it has these products. The CO2 gets given off, that's just waste. But the NADH and FADH2, they want that. And they want to use that for the next thing that they should do. If they know they have oxygen available, what they should be doing then is getting the electron transport chain process running. And so NADH and FADH2 will link in to the electron transport chain process. Okay, so that electron transport chain process is called oxidative phosphorylation. When I say the word oxidative, what do you start thinking about? What does that want? Tend to think about? oxidative kind of molecules do you start thinking about? You think about oxygen when I say the word oxidative? Okay. So in this class, we have to be very, very careful about the word oxidative. Now, in the human and animal phys classes, they probably don't even point this out. And I get it because they don't need to. In this class, we have to make a pretty careful distinction. Oxidative is really referring to the idea of stripping electrons off of something. That's what it's referring to. It's not really dealing with oxygen. But I understand why, in animal phys or human phys, why they don't really talk about it. So I'm going to come back to that idea because we're going to find out that, yes, what I'm going to show you next is the electron transport chain. And it's going to look similar if you've taken animal phys or human phys. It'll look similar to what goes on in our bodies. But microbes can do different versions of electron transport chain that doesn't even involve oxygen. And so then oxidative, if you always think oxidative means oxygen, you get yourself into trouble when I start asking you about these other ways that they can do electron transport. Okay, so that word oxidative, I don't know, circle it, asterisk it, something, but understand that that really doesn't have anything directly to do with oxygen per se. It's this idea of stripping electrons. All right. So essentially, by the time the TCA cycle is done, we started way back with glucose in glycolysis. We took it to pyruvate, put pyruvate into the TCA cycle, all broken down to CO2. That's really good. And then we have NADH, FADH2. Now they're carrying electrons, and their job is to go and give those electrons to something else, some other physiological pathway to allow that other pathway to work. Now, with the oxidative phosphorylation or electron transport chain process, we're going to see that there's going to be a terminal electron acceptor where, like, that's the end of the game. Boom, you made it to the end. Okay, terminal at the very end. So the whole idea of getting that electron transport chain going, you have terminal electron acceptor, and then through that whole process, something's going to get phosphorylated. What do you think is going to get phosphorylated? ADP. All right. So oxidative phosphorylation is another version of how ADP gets phosphorylated. What was the other version? Substrate level. All right. So these are two huge ideas you need to think about. How does ATP get made by microbes? Well, substrate level. You can do that, and they do. Or they could do this, or they could do both. So substrate level phosphorylation, oxidative phosphorylation, two huge ideas on how to get ADP phosphorylated so you can get your ATP. So now in animal phys or human phys, what they often equate as equivalent, they'll say oxidative phosphorylation is aerobic respiration. And I understand that, but again, proceed with caution when you're in this class because it's not always equivalent. Aerobic respiration, aerobic, oxygen is involved. So we're doing that right now in our mitochondria. We have oxygen as our terminal electron acceptor. It's the final molecule in the chain. And so, everything stops there. All the electrons, they'll go to oxygen and the product will be water, which just with the microbe will just excrete that. Doesn't really care about the water. Aerobic respiration is a huge payoff. You get really high ATP yield. As far as we know, having oxygen as your terminal electron acceptor is the best strategy for maximizing ATP. If you can do that, you win the gold star. You are going to survive and do quite well. But we're going to find out that microbes don't always have access to oxygen, but they can still survive. They can still even run an electron transport chain. they'll just stick something else at the end. They just won't have oxygen to deal with. They'll have to use something else. And so we'll see that too. So as far as we know, oxygen is the best electron acceptor on the face of the earth. We don't know of any better when we think about these physiological reactions. And what that means is that will be the best way to generate ATP. You'll get the most ATP if you're using oxygen. So of course, with the microbe, they don't have mitochondria. Mitochondria are ancient microbes, remember? So they don't have internal mitochondria. They have to stick everything on the cell membrane. And so all the molecules involved in the electron transfer chain will be in the cell membrane. And it's just this kind of hot potato game of electrons getting donated to an acceptor and on down the line, just one after the other, passing those electrons until oxygen is reached. So as that's happening, it creates a situation that allows for ATP to be made. And so the fancy word is it conserves some of the energy released during the electron transfer. And some of that energy is used to drive ATP synthesis. So anytime someone starts talking to you about an electron transport chain process in a membrane, you should start looking for a proton gradient because it's almost, oh, I don't know if there is an exception to that. You get electron transport chain, you get a proton gradient. They go hand in hand, can't really separate those two. So let's take a look. So this is a version. Different microbes have different electron transfer chain processes and different players in the game. So this is one example. All right, so here, this is the cell membrane, and you're looking at some big transmembrane complexes that are embedded in the membrane. So they refer to them as complex 1, you see up top. Okay, complex 1, complex 2, complex 3, and complex 4. So those are all transmembrane complexes that are pretty much stationary in the membrane. They're so big that they just kind of stay put. But then you can see that there's some arrows moving amongst those. So you have these other players of the game, like the Q cycle. So that Q molecule, it's a quinone. That is small and mobile. So it can actually associate with complex 1, grab some electrons, move, and go to the next player in the game, donate electrons, okay? And here you see another one, cytochrome C. That's another one that's mobile, small and mobile, so its job is to grab electrons from the donor, accept those, and then donate them on. So the flow going from complex 1 and complex 2, the arrows moving in and coming down all the way to complex 4. Those arrows are the electron movement. And so now you can finally see where the TCA cycle is fitting in, tying in to the electron transport chain. Okay, so you see NADH right here coming in, and you see FADH2 coming in at complex 1 and complex 2. And so those NADH and FADH2, that's coming from TCA. So that's how they link. So NADH says, yeah, I'm fully reduced. I have these lovely electrons. I can come over to the electron transfer chain in the cell membrane, and I can donate electrons into the electron transfer chain. And FADH2 says, yeah, I can do that. I'll do that too at complex 2. And so they put those electrons in, and they keep getting passed along until, in this case, the idea of aerobic respiration there. Oxygen is the terminal electron acceptor. And when it accepts enough electrons, it has to take 4. Okay, it has to have 4 electrons to go from half oxygen to 1 water molecule. So again, this isn't a biochem class. I'm not asking you to know every player in the game, all the enzymes, all that. However, knowing that there are these complexes in the membrane. There's some mobile carriers that help with the electron flow, how it links to the TCA cycle, and what's terminal. It's oxygen in this case. For aerobic respiration, it's oxygen. Now, unfortunately, the artist who drew this really just zoomed in to the electron transport chain and forgot one huge thing that we need to think about, although they're alluding to it here a little bit. So if you look on this, you can see, yeah, okay, there's electron flow going on. And then there's something else going on besides electron flow. What else is moving? Protons. Okay, protons. So what's happening, and I'll get into this later, but basically what's happening as the electrons are flowing from start to finish, we often use the analogy it's flowing energetically downhill, we say. It's going energetically downhill, which what we mean by that is it's creating a situation where there's a little bit of extra energy at key points in this pathway that allows for proton movement across the membrane. So that's what you're seeing here. Protons here in the cell are coming out at key points. So you see protons leaving the cell and going across the membrane to the external environment at key points along the pathway. And it's because of who the players are in the electron transport chain. They're energetics, essentially. So again, trying to show this idea that electron flow is tied to proton pumping across the membrane. And that's really, when you think about it, I mean, the microbe could care less about making water at the end here. That's not its goal. It doesn't care about that. It created and evolved this pathway of protons. That's really what it's trying to do. Doesn't, and so we'll see other microbes do it differently. They evolve slightly different mechanisms, but again, what stays the same is through that electron transport process, you get proton pumping. That's the huge payoff, not getting oxygen reduced to water. That doesn't really matter per se. They don't really care about the water. It's just a game they play to get their needs met of, I want a proton gradient. That's what I want. So if you have protons building up, where you have more on one side of the membrane than the other, you get a proton gradient. Some say an electron motive force, some say electrochemical potential. All means the same thing. Depends on who's talking, how they're going to refer to it. I tend to say proton gradient because it's shorter and it's pretty accurate. Talk about proton, H plus. Okay. So that proton gradient across the membrane is potential energy. You've taken physics, potential energy, kinetic energy, right? So potential energy, we're looking at this idea that once they have a proton gradient, it sets the stage for ATP synthesis. So once they get that, then they're almost set to win the game, the big prize, okay? So the artist should have drawn in this image an ATPase, but they didn't. So this is what they should have drawn in that membrane. Another transmembrane complex, the ATPase, that I'm trying to show here. And trying to show, yeah, there's a proton gradient that got established. And so now, by the laws of diffusion, those protons want to diffuse back across the membrane and go back into the cell, but they can't do it through the hydrophobic portion of the membrane. They're charged. It won't work chemically. So they have to figure out another way to get across the membrane, and so the ATP synthase is the way to go. So as those protons diffuse through, it actually causes those subunits to have a rotational torque force, a swooping arrow, so that you can get rotation And that allows the ADP and inorganic phosphate to physically react with each other and become ATP. That's the big payoff. Not that oxygen got reduced to water. That's just kind of like, I got to play that game so I can get this lovely ATP. So I often think about a lot of these physiological pathways, kind of think of it as like a game they got to play, as long as they understand some of the rules. And then there's always like this grand prize they're going for it, but it might not necessarily be, the end product of these different pathways. You have to kind of look like, okay, what else is going on here that they really, really want to accomplish? So in this case, it's ATP. Questions on that idea? No questions? Well, this is a pretty big concept. So really give this some thought. Again, I'm not asking you to memorize every step in the pathway. I think it's more important. Of course, I'm not a biochemist, so I'm biased that way, but I think it's more important that you understand the big picture idea of what's really going on here and what's really the game, what's the goal, rather than memorizing every single pathway. Okay? I want you to understand the big concepts, because then when whatever class you go on to from next that has any physiology, you get these big concepts. It makes it so much easier to learn some of the other physiological pathways and what's going on with those. And we'll get into some others in this class because microbes do a whole lot more than what we're just talking about here. Okay, I love this image because it's putting together all three things that we just talked about. It's looking at glycolysis and it's looking at the TCA cycle and the electron transport chain all put together. And I thought when I saw this, when I learned all these processes as an undergrad, I learned them in isolation. Like, okay, memorize glycolysis start to finish, memorize TCA, and I never really got how they link together. What's the point? And it took a long time, not until I was a grad student when I started taking microbial physiology classes did it start to finally sink in. I'm hoping maybe I can have this sink in a little bit easier for you now with kind of the big picture ideas that I'm trying to push and not so much the detail. Right? So here you're seeing the glycolysis where you get the little bit of ATP being formed. And that's good. It's not great, but it's good. All right. And then they say, yep, glycolysis goes to pyruvate. And then the microbe says, hey, you know what? I have oxygen. So I'm not going to do stage 3 of glycolysis because I know there's oxygen around me. So I will go and put my pyruvate into the TCA cycle, run that, and I get some more ATP, break down all the way to CO2, my pyruvate, that's great. And then those electrons get transferred to NADH and FADH2. Well, that's really good because then I can shunt those over into the electron transport chain in my membrane. And now I really get a huge payoff, lots of ATP when I do that, because I'm using oxygen as my terminal electron acceptor. And I know that's the best. If I have access to oxygen, I win-win. Turkey dinner, winner, winner, turkey dinner. Okay, it's the best situation. So microbes like E. coli that you've worked with in lab, they do this. But they also have the option. Let's say they're in a broth culture and we're growing them up and we forget about them and they keep growing through days, like say it's spring break. We keep them growing and they're just in this little tiny test tube and eventually they're going to run out of oxygen. Well, microbes like E. coli, they can go, oh darn, yeah, I don't have any oxygen left, but I'm okay because I can stick something else at the end of my electron transfer chain. So I don't know if your lab instructors, they probably haven't got into this, but they will next week, I believe. this idea that if you are able to use something else besides oxygen, you're called a facultative microbe. Facultative. Take it or leave it. If you're a smart E. coli, you will always pick oxygen if it's available. Always, always, always. But there's some times when E. coli is going to be in an environment where oxygen gets depleted. I would love to be a facultative microbe. If I had to be one, I'd pick facultative, because then you can just go, all right, yeah, okay, I don't have oxygen. I'll pick something else besides oxygen that's in my environment. So we'll talk more about that as we get into different physiological pathways. Okay, so that proton gradient that's getting established through the electron transportation process, and then using that to make ATP, that's called oxidative phosphorylation. But the oxidative is really just referring to the idea that you have electrons being donated and accepted, oxidative stripping of electrons onto something, which in this case then gets donated to something on down the line until you get your terminal electron acceptor. So proton gradients are awesome for generating ATP. However, and we tend to focus on that during this phase of the class, but just be aware that the proton gradient can be used for other things by the microbe. Remember the flagella, where there had to be 1,000 protons through the protein to get it to rotate. So they could do that with a proton gradient. They could use it to, like say it's a pathogen in you and you're taking an antibiotic. They might use a proton gradient to get rid of the antibiotic that diffused into their cell. And so there's other ways that they use a proton gradient for sure, but using it to make ATP is really, really good. Questions? All right, so let me just say, you need to spend some time with these concepts. Really solid study time where you're really engaging in the material, really thinking about it. I encourage you to get really silly and do some storylines with what you study. I make up stories all the time of what's happening. I even act it out. You probably notice I move around a lot, wave my arms, and I act out when I would study. I would like pretend I was in a play where I act this all out. And I don't know if it's muscle memory or what, just by that movement, but it seemed to help me on the exam. Not that I got up during the exam. I didn't move around when I was taking the exam, but I could imagine. And so get creative with your studying, especially with this kind of stuff. I know some of you are like, oh, this is so dry. And I get that. But if you have a little fun with it and get silly with it, make stories out of it, it can actually sink in and get into your long-term memory easier that way. So play around with it a little bit, see what you get. And don't get locked into just like, oh, I got to memorize the whole pathway. That's not what I want you to do. I want the big picture ideas to get in your head. Okay? So don't get lost in the detail as you're studying. So we're going to get into more physiology. So that's why I'm really encouraging, what's today, Wednesday? So Friday, we'll get into more physiology, but start studying this stuff now because it's going to get deeper and more varied. So my hope is you're most familiar with what we just covered, that you had it at some point somewhere. So use that to get into this. And then on Friday, we're going to get into more detail on fermentation and other topics. Okay.